Good afternoon, everybody, and welcome to this second section of the ALEA thematic track on artificial life and evolutionary algorithms. My name is Leonardo Vanneschi. I will be the chair of this section. We have 4 talks, each one of half an hour, fuzzily divided into 25 minutes of talk and 5 minutes of questions. Short announcement, I will repeat it at the end. The closing session is at 6 today and not at 6:30. In the program there is a point in which it is 6, which is the global table, and in the detailed program is 6:30, and the right version is the global table. So it is at 6, okay, so we can go with the first talk. The talk is a paper from Fernando Silva, Luis Correia, and Alden Lin Christensen, and the speaker is Fernando. Please.
So, thank you very much for the nice presentation, Leonardo. I'm Fernando. I'm a PhD student at the Instituto de Telecomunicações and at Lab Mag. I work under the supervision of yours truly, Luis Correia, and Anders Lin Christensen, probably the best Danish that ever lived. So let's get on to it. Well, in evolutionary robotics, evolutionary computation techniques are used to automate the design of robotic systems. In particular, these techniques are used to evolve the robot's artificial brain or controller. In multi-robot systems, the evolutionary algorithms are especially important to evolve the robot's collective behavior, in the sense that to evolve a self-organized and structured behavior by hand is quite difficult. So we use evolutionary techniques to do that and to release that burden from the experimenter. So, in evolutionary robotics, the controllers are usually based on artificial neural networks because they're tolerant to noise, like the noise usually present in the robot's sensors. The inputs of the artificial neural network are usually the normalized values of the robot's sensor readings, and then the output values control the robot's actuators or effectors, like the wheels or a gripper. The parameters of the neural network, such as the connection weights or the density, number of, or placement of the neurons, are optimized by an evolutionary algorithm, a process termed neuroevolution. So, in this work, I'll talk about a special kind of neuroevolution, online neuroevolution. And in online neuroevolution, robots learn while they operate in the task environment. So what happens is that there's an evolutionary algorithm that is executed on the robots themselves while they perform their tasks, and this gives them a potential for long-term self-adaptation. This means that if something changes in the environment, like the task requirements or any environmental change, the robots can have this potential to adapt to cope with the new circumstances. For instance, consider this figure here. Suppose that the robots have to catch the ball with the gripper and then take it somewhere else. If the gripper breaks, the robots can learn to move the ball simply by pushing it. So this is the kind of potential that online evolution has, this kind of behavioral modification. Over the last decade, there has been a number of approaches to online neuroevolution in both single and multi-robot systems. What happens is that the dynamics of neuronal models, the fundamental unit of computation, have been largely left unstudied in the evolutionary robotics context. Traditionally, studies use the summing unit that basically does a weighted sum of the inputs. This is a variation of the neuron model as introduced in the 40s by McCulloch and Pitts. But this is more like the studies use it just because it's the standard, although there has been the introduction of different neuron models. For instance, in machine learning, multiplicative units were introduced more than 20 years ago. They're also biologically plausible in the sense that with advances in biology, this kind of multiplicative-like operations have been found in populations of single neurons. A potential advantage that has been almost like enforced into the scientific community in the theoretical studies is that multiplicative-like units can increase the information processing and learning capabilities of neural networks because they may allow for the synthesis of less complex solutions, for instance, neural networks with fewer neurons, and then they can also have a superior computational power. So basically, what we do in this study is that we investigate the dynamics of both summing and multiplicative neurons separately and when combined. That is, we study neural controllers that only use summing neurons, neural controllers that only use multiplicative units, and then controllers that use the both types of neurons. We used the standard summing unit model, that is, as I mentioned, a neuron that performs a weighted sum of the inputs, and we used the general product unit model, in which the inputs are multiplied after being raised to the connection weight. We conducted a series of simulation-based experiments. We used the model of the EPIA robot. The EPIA is a small, 75 millimeters in diameter, differential drive robot. It has short-range sensors up to 25 centimeters for detection of obstacles or for transmitting any information. And it's also a bit slow as it moves up to 13 centimeters per second. So this is the robot model that we used. We studied a standard task in evolutionary robotics, an integrated navigation and obstacle avoidance task. What happens is that in this task, robots have to move as fast as possible, as straight as possible, and then they have to avoid obstacles. So there's a trade-off between maintaining speed and forward movement and avoiding the obstacles. This task is usually studied in single robot systems. What we do here is, as we have a multi-robot system, each robot is an additional and moving obstacle. We used two types of environments. This one we called the plane environment. It's basically a squared arena surrounded by walls. And we studied a more complex environment that has obstacles. The obstacles are made of the same material as walls. And they intuitively make it harder for the robots to learn the appropriate behavior. I'm not going to go into much details about the underlying neural evolution algorithm. It's called ODNIT. Basically, ODNIT is an online neural evolution algorithm that implements the evolutionary process according to a physically distributed ideal model. This means that each robot that is executed on ODNIT has an internal population of candidate solutions to the task, and then robots probabilistically broadcast a possible solution to the task to other robots in the immediate neighborhood. So it's really a physically distributed island model. ODNIT has some features that are similar to those presented by NIT, by Kenneth Stanley this morning, in the sense that it's a special neural evolution algorithm that evolves both the neural network's weights and topology. In particular, the population stored by each robot is composed by networks that have no hidden neurons and that start with each input connected to every other output neuron. Then, as evolution takes place, new neurons and new connections are added through mutation. In particular, a new node is added by splitting a connection, and a new connection is added by connecting to previously unconnected neurons, and these neurons are chosen randomly. In order to assess the effects, the potential benefits, the advantages and disadvantages of different neuronal models, each time a new neuron is added, we conducted three sets of experiments. Each time a new neuron is added, in one set of experiments, the neuron is always a summing unit. In the second set of experiments, the neuron is always a multiplicative neuron. And in the third set of experiments, we randomly assign either a summing or multiplicative role. This means that in this hybrid setup, we can have networks that perform a weighted sum of the inputs, a weighted sum of products, for instance, or that perform the product of weighted sums. So it's a bit more flexible in terms of the computation they can perform. So ultimately, online evolution evolves the robot's behavior. We wanted to have a means to systematically categorize and classify the robot's behavior. To that end, we used the generic Hamming distance-based behavioral metric. It was studied by Mouret and Doncieux in a recent contribution, and this measure is based on the mapping between sensors and effectors. We have a behavior is defined as a set of T observations measured consecutively throughout time, and each observation is the normalized values of the sensor readings and the actuation values. They are transformed into a set of bits. In particular, if the value is greater than 0.5, it becomes a bit 1. If the value is lower than 0.5, it becomes a bit 0. So one observation of behavior is something like this. Based on the process I previously described, we used two measures to classify the robot's behavior. One of them we call the inter-behavior distance. It's basically a distance between two behaviors. So we have the set of observations, and the distance is the summing of the pairwise distance between observations at a given time. The distance measure we used here is the Hamming distance. So we have two bits that are different, and at this time the distance is 2. Here in this observation we have a bit that is different, so we have a Hamming distance of 1, here a Hamming distance of 3, and then the total interbehavior distance is 6. That is the sum of all the distances of all observations. We also wanted to see, more than comparing two different behaviors, or more than seeing if two behaviors are different, we wanted to analyze how much a behavior varies during task execution. So we used the same process. We defined the intra-behavior distance that, as I said, describes how much a behavior varies during task execution. The principle is the same, but instead of measuring differences between two instances of the behavior, the differences are measured between the same instance and using the consecutive observations. So in terms of the comparison of performance between the different neuronal models, we use the number of evaluations, that is the average number of controllers that each robot tests before a solution to the task is found. We also analyze the complexity of solutions, that is how many neurons and connections are added to each network capable of solving the task. And we also analyze the generalization capabilities of each controller. That is, if we restart the task and we don't allow further evolution, how many controllers are still able to solve the task? That is, how many controllers are robust enough to be redeployed and still be able to do what they usually did. So we tested 3 types of controllers: those that use summing neurons, those that use multiplicative neurons, and the hybrid controllers in the 2 environments. And in each experimental configuration, we conducted 30 independent runs. In terms of the comparison of evaluations, the results were actually a bit surprising, but still interesting. And the main results are the fact that controllers that use summing neurons require an approximate number of evaluations that appears to be independent of the task complexity. That is, in the plain environment, the environment without obstacles, and then in the complex environment, there's an approximate number of controllers tested before a solution is evolved. On the other hand, controllers that have a multiplicative component, either by relying on multiplicative neurons alone or by relying on the 2 types of neurons, appear to be affected by the increase in task complexity. That is, we change the environment is harder, and then evolution requires more evaluations to synthesize a solution for the task. So we thought, okay, these types of controllers require more evaluations. Let's see how this reflects on the topological complexity. Counterintuitively, we saw that multiplicative neurons and multiplicative controllers, and then the hybrid controllers, are usually the multiplicative controllers are usually less complex than the other 2 types of controllers. This is actually a bit surprising, as I said, because of the number of evaluations. And we analyzed this thoroughly, and we saw that because the multiplicative controllers rely on the connection weights alone, they need for the connection weights to be finer, to have a finer grained adjustment. That is, they have to be almost tuned to detail in order to perform the computation reliably. Hybrid controllers also have this tendency, but more importantly, they usually require this intermediate number of evaluations. But there's an important part that we weren't aware when we started the study, and it's actually an important aspect, is that we're using different types of neurons. So when we're recombining networks, we are mixing networks that have 2 types of neurons, and this may very well damage the computation that the network performs. So this higher number of evaluations with respect to the summing neurons, and then the higher complexity is due to the fact that in order to minimize the effects of recombining different networks, more structure has to be added to compensate for it. Also, a nice result is the fact that all the controllers, either using summing, multiplicative, or hybrid neurons, are high-scoring controllers. That is, the average fitness varies from approximately 91 to 95 out of 100. So they're usually high-scoring controllers. There's no significant difference here So, as I said, we also conducted a series of tests to analyze the generalization performance. For each group of robots capable of solving the task, we restarted the task 100 times. That is, we redeployed the robots in random positions. And in the complex environment, we also placed the obstacles in random positions. What happened is that in the plane environment, because it's an easier environment, all the types of controllers have a quite good performance. That is, they still continue to solve the task regardless of being redeployed. When we shift to the complex environment, the performance drops significantly, except for the hybrid neurons. So these hybrid controllers display a high generalization performance in the sense that they appear to be more robust to changes in the environment, to redeployments, and can still solve the task without additional evolution. Actually, this result is quite important in the sense that for online evolution, it means that robots using hybrid controllers can have a more efficient adaptation to different contexts without additional evolution. That is, you have a controller that is robust enough to cope with some changes in the environment. So, as you probably noticed, the results were not fully consistent across all the different setups because we had controllers that required more evaluations but then were less complex and things like that. So we wanted to see how did the evolutionary search proceed through the high-dimensional genotypic space. To that end, we used Salmon's nonlinear mapping to analyze all the solutions that lead to the final solution, so the stepping stones, as Kenneth Stanley said this morning. Here I have plotted the most different genotypes, and I also have to abstract this process. Basically, we had a high-dimensional genotypic measure, as used by ODNIT, and we reduced the distance to, we made a dimensionality reduction to this Euclidean space, and this means that each point in this graph is a genotype, is a network. So networks that are close here means that they are topologically similar. Networks that are far away from each other are topologically different. Generally speaking, this kind of results showed us that there's a balanced exploration of the search space. That is, regardless of we using different types of neurons, there's the fitness landscape is not significantly altered. That is, the different types of neurons don't pull the search into any specific direction. So this raised the hypothesis that if the search is similar, then it may very well be that the dynamics of the neuronal models cause for the robots to evolve different behaviors. We analyzed this hypothesis using the distances I introduced previously, and the results basically tell us that, or they appear to tell us that some neurons during the task are able to, on one hand, they evolve more diverse behaviors, and then these behaviors also change more during task execution. Multiplicative neurons, on the other hand, appear to evolve more static behavior, more robust behaviors that change less comparatively to summing neurons during task execution. Interestingly enough, hybrid controllers appear to have this hybrid behavioral characteristic in the sense that either they vary or they evolve very different behaviors, or they evolve intermediate behaviors that can vary more or less depending on the task complexity. So to summarize, we analyzed the dynamics of summing and of multiplicative neuronal models. The hybrid controllers are competitive in the sense that they require a competitive number of evaluations. All the controllers are high scoring, including the hybrid controllers. And more importantly, the hybrid controllers yield a superior generalization performance. So in terms of future work, we have seen that hybrid controllers can balance between maintaining and modifying the behavior. This appears to depend on the task requirements and complexity. However, we need an additional analysis and more experiments to see if there's a trend in the results or if it's just due to this type of tasks. In particular, we intend to conduct an analysis of the neural activation in structural patterns, that is, to see, for instance, if evolution uses multiplicative neurons or and summing neurons in any specific way that allows for these behavioral characteristics. And afterwards, we intend to study macroscopic properties, that is, to study how to evolve modular networks that are regular and hierarchical, and in that way to see if both the microscopic properties, like the type of neuron, and the macroscopic properties facilitate online evolution in real-world complex tasks, that is, online evolution in real robots. Well, and that's it. I rest my case.
Does it work? Yes. Thank you very much, Fernando. We are in perfect timing, so we have 5 minutes for questions. Any question? Please.
So, thank you for your presentation. I was wondering, you're exploring cooperative behaviors in a sense that the robots help each other evolve solutions for environments with one particular task, right? They all do the same thing.
Yeah, they all do the same thing, but depends on what you mean by cooperative, because it's a collective task, but they exchange genetic material, but that doesn't mean it's cooperative.
Yeah, that was my point. What would you think it would be necessary to create cooperative behaviors?
Well,
in which you had more than one task. And you needed cooperation in order to, and you needed diversity to kind of tackle different problems.
You mean diversity in terms of the behaviors, that is?
In terms of behaviors,
some
yes.
kind of task specialization in,
yeah,
or something like that. Well, one of the things with online evolution is that it can easily specialize the robot's behavior. For instance, if you're in a different part of the environment, you can, for instance, recognize that you're in a different part of the environment and try to evolve a different behavior by relying, for instance, on a different fitness function. So that's one way of doing it. You can also, also an advantage of online evolution that each robot doesn't have to execute the same evolutionary algorithm. So if you want different tasks, you may split the group and say, okay, this part of the group tries to do this part of the group tries to do that, but in that case you're defining what the group has to do. That's also
what if you don't know the kind of the prerequisites to accomplish something, and you have multiple things that need to be done differently, and if in cases in which the task does not depend on the position of the robots,
then you use novelty search. No, I'm kidding. No, it depends, because aside from using novelty, you need a fitness function. You need to tell the robots, okay, this is what you're going to do, this is what you have to do. If you have a general fitness function, a high-level fitness function, then you don't have to specify what the robot has to do in detail, so they may do it in different ways. But if you really want them to do different tasks, then you have to come up with some way of using different fitness functions, or like really using or trying to evolve some kind of task specialization.
Yeah, because I was imagining that the diffusion of the partial solutions would be damaging to the behavior diversity that you would require.
It depends. Actually, that's why we're trying to study how to evolve modular neural networks, that you can have subcomponents of the behavior that are common between the two tasks. So you can reuse something that was evolved by other robot, even though your robot is trying to solve a different task.
Okay. Thank you.
Hello. Hello. Thank you for a very nice talk and very interesting. I would like just to ask a short question. What about architecture for your neural network? Have you studied the effect of restricting the architecture just to some kinds of networks, such as a feed-forward, even if it is an Hellman recursive network or something?
Yeah, actually, I didn't mention it but evolution can add any kind of connection. So here, the example is a feed-forward network, but evolution can evolve recurrent networks or whatever. So I haven't paid attention to that because it's usually, at least in my opinion, it's usually a minor thing.
Well, because you were checking in the graphics, you see a huge number,
the
huge
complex
differences. due to the connections, not to the units sometimes.
Mhm.
Of course, the units are needed for the connection.
So it looks like the hybrid has some appealing qualities compared to the not hybrid, but do you have a feeling for like what other hybridization should there be then? I mean, is it just multiplicative and summative, or is it like there's some other things that would even add even more important abilities there?
Yeah, actually, I studied some different neuronal models also, and one type of neurons that I think can be useful is those related to learning. Like, for instance, if you use neuromodulation, you have that special type of neurons that can control how the other neurons react to certain inputs. So neurons that basically, special neurons that basically are able to change the weight of the connections of other types of neurons. This is not the most correct example, but that's the main principle. So that's also another type of neurons that can be important for evolution and learning, really, for online learning.
Okay, so we can thank the speaker.
The second talk is a presentation of a paper by Rui Lopes and Ernesto Costa, evolving an harmonic number generator with REN code. And the speaker is Rui.
You tricked me. So hello again. My name is Rui Lopes. I'm presenting experimental work with the regulatory network computational device aimed at find the implications of some design alternatives of the underlying genotypic representation. Before everything, a little bit of context. We know that representations, or most representations in evolutionary computation, are still very attached to old notions of biology, so from the beginning of the field. Nowadays, we know there are a plethora of processes, of different processes, that are part of natural systems. And researchers still tend, most of them, to disregard these processes. So what we are trying to do is to introduce regulatory processes into evolutionary computation representations. In this case, we start with an artificial regulatory network model, and from it, we try to extract an executable graph. This has been presented before, and it has been applied to the problems like the pole balancing, symbolic regression. In this case, it was the causes polynomial that was tackled, and the Santa Fe ant trail. More than that, we extended this representation to use also feedback connections that would allow to tackle problems with recursion. Examples are the squares regression, Fibonacci sequence generator, and also streaming and bit parity checker. Today, the goal is to study the influence of design alternatives of the underlying artificial regulatory network model. These are the initialization of the genome, which can be either random or by duplication and mutation, according to the original proposal, the size of the genome, and third, the codification of the genes, whether they overlap in the genome or they do not overlap. Before going into RAN code, we need to introduce the artificial regulatory network model as it was presented by Wolfgang Benzav. So the genome is a bit string where we can identify genes by a promoter. This promoter is a fixed sequence of 8 bits, but in the total it has 32 bits. Then upstream of the promoter we have 2 regulatory sites, one for excitation and the other one for inhibition. The rest is a coding part of the gene. It is composed of 5 32-bit chunks, and this will code for a protein. So how does this code for a protein? We apply a majority rule over the set of the 5 32-bit chunks, obtaining a protein with a signature of 32 bits. So we go each position of the chunks and we apply majority rule, and with that we obtain the resulting bit for the protein's signature. With this, we can build a network where proteins then bind to the regulatory sites of each gene. How is this done? This is done by applying an exclusive OR between the bits of the protein signature and the bits of each of the regulatory sites. This can then vary between 0 and 32, obviously. Finally, there is a binding threshold, usually set to half the way between the 0 and 32, so to 16. This is derived from other works that were presented by other researchers that studied networks and the properties of the networks generated by this model. And this is used to discard connections. So each protein that has a connection to a regulatory site that is weaker than this binding threshold will be then discarded. Now, RAN code. In RAN code, we use this artificial regulatory network model as the genotypic representation to extract the executable graph. It is a 2-step process. The first, which is to go from the artificial regulatory network, which is composed of multiple excitation and inhibition connections between every gene, has to be reduced in order to reduce the amount of information that we are dealing with and also to remove cycles from this graph. This is done with a simple algorithm which consists in taking each pair of connections, inhibition and excitation, and transforming it into only one connection equivalent to the difference between both of them. In this step, if a connection goes below 0, it is also discarded. So we consider it's not so relevant. Finally, we then go to each pair of genes, and if there are both connections from gene A to B and from B to A, we take only the connection which is the strongest. The second step, to extract the executable graph from this reduced graph, has a bit more complex algorithm, but the idea is to choose the node which has the most input connections, and then recursively from this node we build the executable graph until we reach the leaves. The leaves, nodes without input edges from any other gene, will correspond to the terminal nodes. There is one detail still to know, how to map a node into a function. Here again, we apply the majority rule over the protein signature according to the size of the function set. In this example, if you have a function set of size 4, and we apply the majority rule and we obtain, for instance, 1, 1 in binary, this will correspond to 3 in decimal base, which means that we'll take the last of the functions in the function set as the mapping to that node. Well, this said, I will introduce the problem that we tackle this time. It is a well-accepted benchmark from the papers that were published last year on GP benchmarks. We chose only one, in this case the harmonic curve. It is given by this function that is presented on the screen, and it consists of two tasks, one of interpolation and another one of extrapolation. So we first evolve an approximation, and then we test it for generalization over an extended set of points. In this case, we evolve with 50 points, and then we test for generalization over 120 points. The function set is the one presented here that is usually presented in the literature. The reciprocal sum is used, which corresponds to the sum of the reciprocals of each input. It substitutes protected division, but it still has to be protected, of course, because if we get a 0 on the reciprocals, we have to return some other value. In this case, we return a very big value so that it will probably be an invalid individual. The terminal set is simply the n that is used. No? There are no constants. We didn't use any constant. The fitness measure we used was the mean squared error. And we halted the runs whenever this error came below 10 to the power of -3. Excuse me. Now the experimental setup. So as I told before, we experimented to know the influence of 3 different design alternatives in the underlying model. This is the initialization of the genome, which was presented initially as a duplication and mutation process. Maybe I skipped this part before. Duplication and mutation means that we start with a seed of 32 bits, and then we duplicate the seed and mutate, we duplicate and mutate, and so on, according to the desired number of these events. So we counteracted this with the random generation. We simply generate randomly all the bits for the genome. The other aspect is whether genes may overlap or not. In the original model, it was only mentioned that promoters could not overlap, and this we maintain. And then with the talks with other researchers working on this, I found out that actually they also do not overlap the genes, which was something that we were doing. So we tested both to see whether there is influence of this aspect or not. Finally, we tried 5 different sizes from 1024 bits up to 16,364, which corresponds to the number of DMs of duplication and mutation events that you have below. So from 5 to 9. We used a 10 plus 100 evolutionary strategy. So we start with an initial population of 100. We have a maximum number of evaluations allowed to 1 million, but we halt the run if it reaches a mean squared error below 10 to the power -3. And for each of the variants, we did 100 runs. Given this, we can go to the results. Here I present 2 circuits that reached mean squared error of approximately 10 to the power of -5. I present these 2 solutions because, well, it is obvious that the natural logarithm will appear in many solutions, since it's already a very good approximation to the function. But we could also obtain other circuits that have, for instance, multiplication at the top, although they are not so common as the other ones. This is the plot of a target function versus the sample individual. As you can see, the error is quite low. They are almost one over the other. And now the analysis of the results. As we can see here, the effort taken to find a solution was much lower in the random experience. Below you have whether it was used duplication and mutation, it has dm, and the corresponding size, so from 5 to 9 as presented before. Or you have the RND, which corresponds to random initialization, again with 5 to 9 for the size. And finally, if the genes do not overlap, we have an F at the end of the label. As you can see, there is one experiment missing. It is two experiments missing. It is both duplication and mutation and random initialization with size 5 and with no overlapping genes. These are missing because the experiments could not reach a solution, since most of the times the individuals were invalid. They had too few or none or any protein, because the genome was so small and the genes could not overlap. Now, we did a statistical analysis of these results. We used non-parametric tests, since the results don't follow a normal distribution. We applied the Kruskal-Wallis to be sure there was significance first, and then we solved the pairwise comparisons with the Mann-Whitney-Wilcoxon test, applying the von Ferronic correction. This is pretty standard analysis for this type of results. Here is a summary of what we could analyze. So we present a plus if the experiment in the row performed better than the experiment in the column. That is, if the number of evaluations taken to find a solution was less than the one in the column. We present a minus otherwise, and if there was no significant difference, then we present a tilde. As we can see, mostly all the random experiments are significantly better than the ones with the duplication and mutation. Please do not understand me wrong. This is not random search. There is still evolution. It's just the initial generation of the genome. It is random or using duplication and mutation. Now to the generalization task. I present here the error bars for the generalization error, where we can see, again, now we have numbers. I could not use the labels here. But in this side we have duplication and mutation. On that side we have random initialization. And as we can see, random initialization also achieves a better generalization performance. Now the success rates, not every experiment could generalize below mean squared error of 10 to the power of -3. But as we can see, in the case of the random experiments, it was quite close for most of them. We tried to find some type of explanation for this, whether it would be in the number of proteins or functions coded in the genome. That's presented here, the number of proteins and functions. In both of them, we have less functions than proteins, which is normal, but this difference is a bit bigger with the DM. Although we could not find a correlation with the generalization results. Here you have the generalization error with the number of proteins. There is a slight correlation. With less proteins tend to have better results, but we cannot deduce this, cannot infer this. So, summarizing, the size of the genome does not influence much the performance. There is some influence, but not that much. In general, we prefer small genomes. Also, experiments will run faster with smaller genomes than the bigger ones. Then, overlapping genes also did not have a significant difference in the results. So whether they overlap or not, as long as the promoters do not overlap, it will be okay. And of course, the most important aspect is the initialization procedure. Here we found that the random initialization is better than DM, performs better. It was not the original proposal, although other researchers have shown with a model based also on the ARN, have shown before that also in their case they obtained better results with random generated genomes than with duplication and mutation events. And just before ending, and to tease you a bit, because I think we still have plenty of time, instead of presenting a slide called future work, I will just present what is going on now with RunCode. We are trying to apply the extended model of the artificial regulatory network, which has a distinction in the promoters, allowing to code for both transcription factors and products. And also, they have hard-coded inputs. In this case, they used 4 proteins. We could use even more if we wanted. And with this, we want to be able to solve problems where multiple outputs are needed. And in this case, I present here a full header that was reached. This is still very preliminary work, but is giving its fruits. And with this, now, I end my presentation, and thank you all for being awake.
Thank you. We have time for questions. Please, Wish.
Gene regulatory networks are inspired in biological genomes, right? They are inspired in biological genomes. As far as I understand it, in biology, gene regulatory networks are important because organisms have complex development, growing, etc., etc. And my question is, it seems that you have applied that to problems where this feature is not really needed, and just to solve some more or less complex functions. I would say less complex functions, and correct me if I'm wrong again, but the one that you referred, that you had tested, the running parity test or something, it's something that you need a 1-bit memory. Is it more complex than that? So it seems very simple problems where there is not much development of the phenotype. And so I would like to hear your comments on, or maybe I'm completely wrong, or what is your idea on this?
That's an interesting comment. You are not completely wrong. What you said about about the gene regulatory networks, it's very true. And this model that tries to replicate that behavior, and actually Banzaf was successful at doing that, at replicating, well, at least some aspects of the dynamics of protein concentration variation along time. He was successful with that. And this is a part that, if you noticed, I didn't introduce in the presentation. So there was a dynamic part of the ARN that was left aside, precisely because in this model we only use the network itself to extract executable graphs. It's very similar, let's say, to GraphGP, although we use a linear genome. There is no positional dependence, for instance. And so it is adequate to this type of problem, as there is no dynamic simulation of the ARN. In the case of, for instance, the pole balancing, the model has been used with the dynamic part by Miguel Nicolau, to solve the pole balancing, so to evolve controllers for the pole balancing. In this case, what we did was to regress a function, a controller for the pole balancing. So not a dynamic one, it's a fixed structure. It takes the 4 inputs, gives you an output, whether to the left or to the right, and it's as simple as this. So there is no dynamic part here. Did I answer your question?
Yeah, but if I may, partially in the sense that I wonder why would we need something that is seemingly more complex than, for instance, GP to solve these problems, and what have you gained? Because I didn't see any comparison of this towards GP in terms of complexity of evolution, number of generations, length of the genome, etc., etc.
We have done that in the past. We obtained better success rates, for instance, although there were no direct comparisons. We obtained better generalization results, for instance, in the pole balancing. In this case, I'm focusing on the pole balancing because there is a direct comparison. The best that was obtained until our work in generalization was success in 422 starting positions out of 625. And we increased this to 495 positions out of these 625. So here you have one of the advantages, for instance. In terms of number of evaluations, I don't know if it is bigger or smaller than typical GP, but I know, for instance, that Cartesian genetic programming has even bigger numbers of evaluations to solve the same type of problems, the original proposal. And just to add a bit more, in terms of development of natural systems, we are now trying another approach, but we are using a different mapping, we are using grammatical evolution, and in that case, we are simulating the network so that the system automatically defines the order of the proteins, so the order of the codons that are used in grammatical evolution. It was work presented this year at GECO also.
Tell us more about the multiple output part, because it's so important for GP. It's what is missing in normal GP systems.
We presented also in GECO preliminary work in this sense. So we developed this RAN code version 2, let's say. I took the chance also to simplify the second algorithm that I presented from the reduced graph to the executable graph. This one is a bit messier than the new one. And besides this, we tested the new approach with an old problem, the pole balancing actually, and also we presented just very preliminary results in creativity. There was nothing new added to the state of art of creativity. We simply made generators of RGB pixels. So we have images, some of them actually that look a bit like ones Stanley presented in the morning, not the evolved ones, but the messy ones. And it gave interesting results. Now for the full adder and the multiplier is a bit harder. This one that I presented here at the end, for now it was obtained 1 in 30 experiments. We still have to try to find why, and you still have to find actually a multiplier. I still could not do it. But the idea is to apply it to full adder, full multiplier, and so on. Try to increase the difficulty also. Like more bits to the adder and more bits to the multiplier.
I wanted to say something about this also. I don't want to prevaricate. We have time for other questions, but I am also interested in this multi-output model, which is actually, I think, one of the most interesting open issues of this kind of study. Absolutely without knowing the answer, I was wondering if every step that you have presented works also in the case of multi-output, and I was thinking in particular to the reduction of the graph. I mean, the reduced graph has to have the same input-output behavior as the original one. And can you apply the same algorithm if you have more than one output?
Yes, if I understood correctly your question, so the answer is here. This model, you have the products coded in the genome. So you can have from 0 actually to many of them. What we do is, if you don't have, you consider it invalid individual. From the point you have, if you have more than you need, you discard the other ones. But then the algorithm works pretty much the same way. You take the output 1, output 2, and then you start building according to the connections of the other genes to those outputs. It is important to notice that in this kind of model, You don't have connections towards the inputs. They are extra proteins that are added. Nor you have connections from the outputs to the other genes, because they are just outputs. They are not regulatory proteins. So only the transcription factors connect inputs and outputs. Thank you.
Let's pass to the next talk. The authors of the next paper are Roberto Ugolati, Pablo Mesejo, Joseph Nashed, and Stefano Cagnoni. And the presenter is Stefano.
Okay, thank you, Leonardo. I'm going to present a work which is somehow summarizes the work we have been doing in the last 3 years, and especially during the MIBISOC project, in which Pablo and Youssef have been directly involved as early stage researchers, and Roberto Ugolotti as an additional local PhD. The MIBISOC problem, the MIBISOC project was related to developing soft computing methods for image analysis in the medical field. And to do so, we had to develop algorithms that were particularly efficient. So we spent a lot of our time developing GPU versions of meteoristics. And as a medical application, one of the medical applications we considered was the recognition of the hippocampus in microscopy images of the brain. And so we somehow put it all together. We have developed, as we will see, we have developed a library including, for example, PSO NDE implemented on GPU, on CUDA. And on the other side, we have developed a model-based algorithm for optimizing elastic models to make the recognition of anatomical structures. So what I'm going to talk about today is somehow a study that puts together everything we have done by trying to optimize algorithm parameters using meta optimizers that are in turn evolutionary algorithms. In particular, we are optimizing the parameters of the differential evolution using differential evolution and particle swarm optimization. And we are testing our approach using standard benchmark functions, so let's say toy problems, and the real application we studied, which is the recognition of the localization of the hippocampus in in the microscopy images. So, of course, it's widely known that parameters may have a large influence on evolutionary algorithms. So there are two possible approaches to optimizing parameters of an algorithm. Let's say an offline parameter tuning, in which you choose the parameters, then you run before the optimization starts, and then you run the algorithm and see what happens. Or you have parameter control, which is, let's say, an online process by which you control, you change dynamically the parameters during the running of the algorithm. But we are considering the first time, the first kind of approach, the simpler parameter tuning. Of course, there are a lot of issues, as we will see in the end. There will be fewer conclusions than open issues. And parameters are usually not easy to choose because, of course, the behavior of an evolutionary algorithm depends on the problem it has to solve, depends on the fitness landscape of it has to explore. Results cannot be immediate as evolutionary algorithms are stochastic algorithms and so on. And of course, manual tuning of the parameters has a lot of drawbacks because it's biased from some possible misleading ideas that the programmer may have. It may be tedious and time consuming when you don't know exactly what you are dealing with, the problem we are dealing with. It's a matter of trial and test, and usually it's not easy and quite lengthy to get to parameters that produce an optimization that satisfies you completely. On the other hand, automatic tuning is, of course, unbiased. It can perform, especially in the case we are considering, in which we have implemented quite very efficient versions of the optimization tools, a huge number of tests, and it can explore, let's say, novel solution, novel sets of parameters. Of course, when you talk about meta-optimizers, we can quite obviously we are considering optimization algorithms that are used to optimize the parameter of another optimization algorithm, which is something like this. So what we did is we considered D, the parameters that regulate D, and we used on top of it either particle swarm optimization or differential evolution to optimize the parameters for some specific problems we have considered. So the encoding we used, we have parameters to optimize that are both real parameters and nominal parameters, and on the other hand we have parameters, we have methods that are that use real coded genomes. So we had to define some sort of simple encoding for the nominal parameters using the real numbers. So, for example, for crossover and mutation, for the choice of crossover and mutation, in which we have a binary choice for crossover and 3 different kinds of mutations, we used as many real numbers, and we chose the kind which was assigned the highest value. So we had an homogeneous genome of all of 8 real numbers, by which we could encode 5 parameters, 3 of which were real, and the other 2 were nominal parameters. Of course, so each particle or each element in the population encodes the fitness encodes an algorithm. A set of parameters that regulates an optimization algorithm. So the fitness of the individual is the average fitness obtained by running several times the algorithm on the problem and to see how its behavior and how it works. Okay, the key point of this presentation is mainly the fact that there's nothing really new in this approach. It's already been attempted several times. There are open issues, but that using CUDA, using a GPU implemented versions of the algorithm, we could actually do quite in relative short times, we could do quite an extensive set of tests, which allowed us to take some, at least to get some results which can be discussed, if not really final conclusions. And the use of GPU, of course, for parallelization is particularly effective in the case of evolutionary algorithm in which there is an intrinsic parallelism which is quite marked. And in fact, GPU works particularly well when you have the same code executed independently on different data. You have the same behavior for each thread, and so if there are the fewest possible branching instructions, and especially if you can make the fewest possible accesses to the global memory, which means that you can run, obviously, independently the different threads. We have made an implementation of a few meteoristics, and we have developed libcuda optimize, which contains, in particular, an implementation of DE and PSO, and can be freely downloaded from that address. In the end, PSO and DE share a very similar structure, so we could develop 3 kernels, which were just the same architecture for both algorithms, which is one kernel for particle updates or for the updating of the values of the population, the fitness evaluation, and selection. We can use the same. The first and the third kernel would be exactly the same for meta-optimizers and optimizers, and the second kernel would be an optimizer. In case of the optimizer will be a function evaluation. In case of the meta-optimizer, the fitness value is obtained by running an optimizer for several times. So it is actually a full evolutionary process. Just to give an idea, of course, even in this case, everything is very dependent on population size, number of evaluations, kind of problem, and so on. But just to give an idea, and of course the hardware on which the comparison is made, we compared the result of optimizing rastering function and finding the best DE parameters for optimizing the rastering function on an NVIDIA card versus, let's say, traditional Intel multi-core processor, in which we didn't use the parallel features. So we just run a single thread. We just develop a sequential single thread version in C++ of the, and we got a speed up of about 6 to 8 times. Depending, of course, on the dimensions. The larger the problem, the larger also the speed up. So it was about 6 times for 16 dimensions and 8 for 64 dimensions. We used 2 different test suits. We considered first for classical benchmark functions, which are the Zakharov, Schwefel 1.2, Rastrigin, and Rosenbrock. And then we compared the parameter choice with the results of a systematic search of the parameters space. So we at least did a very calculated a ground truth for our algorithm by sampling quite densely the space of parameters for these 4 cases, and we compared the result obtained by systematic search with the results obtained by the by parameter optimization. And then we consider also the real problem we were considering in the MIBISOC project, which was hippocampus localization in histological images. So we had as ground truth the sampling of the parameter space. We consider more than 13,000 points, which means we ran, I think, 10 evaluations of the algorithm with each set of parameters for 13,000, more than 13,000 times, and then we compare the results we obtained with 5 rounds of our meta D and meta PSO to optimize the parameters of D. The parameters of the meta optimizers were chosen after considering the results of a paper we presented at GECO, in which we considered the result obtained on 8 benchmark functions to determine parameters which seemed to generalize quite well over these functions. We considered 24 as population size and 64 generations. So we considered in the end for each run about 1,500 parameter sets. And we considered the parameters we actually used are listed in the last 2 lines for DE and PSO. These were the results of the exhaustive search, and we considered even here we saw that there are functions with a lot of local minima. So even in searching the parameters, so we consider the range of the 10 best solutions, which are in any case rather limited. And we compared the result of the ranges found in the 5 runs of our meta optimizer. As you can see, the parameters are more or less the same, so there was a good, satisfactory results. The parameters that were chosen were very similar, or were more or less in the same range as the one that were found by exhaustive search. The actual the real-world problem we considered was hippocampus localization in histological images. As you can see, even if the shape is roughly the same in all the images, the features of the images can be very different. And the most invariant feature is the presence of those 2, let's say, lines, which are 2 particular structures of the hippocampus that can be, which we actually used to create a model that could allow us to locate the hippocampus in every image, whatever the deformation of the appearance of the contrast in the image was. So the model was based on a deformable model, which could and the goal was to make the model, to overlap the model to the actual image as much as possible. So we have some parameters that are that describe the model that can be varied and adapted to match the actual appearance of the hippocampus of the model with respect to the actual appearance of the image. Mainly, we considered a set of points, one one set models exactly the 2 lines and must have a darker appearance, and there is another set which is immediately located immediately outside the 2 lines that must have lighter values. The other consideration is how much the model we consider is similar and matches as shape the hippocampus. So we are considering what is usually called in the deformable models terminology the inner energy, which is the actual deformation we have to apply with respect to the basic model, which is the average, the prototype of the hippocampus, and what is called the external energy, which is the energy which corresponds to the similarity between the image features, especially the color or the contrast of the image. So in the end, there are two terms, an external term E and two other terms which correspond to these two different criteria. So we must somehow we must found the deformations in which of minimal energy that corresponds to the maximum overlap between our model and the hippocampus to detect the area in which the hippocampus is actually present. The results we obtained here, we didn't report in the standard deviation of the fitness, which had to be maximized in this case in the results below, but the statistical tests showed that the automatic parameters always outperformed the manual parameters we could find that were already quite good as we used them to in the paper that was published on pattern recognition letters. So actually we could improve on already quite good performances we had already got. So the conclusions are we used a method to find the best parameters for NEA that considers parameter tuning as in turn as an optimization problems, and so uses other evolutionary algorithms to find the best configuration of D, which is another meteoristics. We tested it on benchmark function and real-world problem with good results, but the main thing was that we could perform quite a huge amount of tests in a reasonable time thanks to its GPU-based implementation. There are, of course, a lot of issues on the problem, which was probably not even the very center of our interest, but it was mainly an application that required a lot of computation time and efficient algorithm. So the problem of how to, of the search for good parameters for the meta optimizer, you could go on and on recursively infinite times, of course, but what seems to happen is that as you climb the levels of the meta optimizer, the fitness landscape tends to become smoother, and we actually are finding out that probably many times it's not necessary to use a very complicated optimization algorithm to set the parameters, and you can get quite good results. We could get quite good results on some of these functions, even with a very simple hill climbing algorithm. Then, of course, there are the problems related to the fact that all the performance of an evolution algorithm is definitely problem dependent, and also that we are considering only the solution quality, but actually we would like probably to consider other criteria in optimization, such as the speed which is needed to reach a solution, or the robustness of the algorithm. So we could even consider to go to consider this a multi-objective fitness problem. Thank you.
Thank you, Stefano. Any questions? Okay, it is always embarrassing to ask questions to a friend, but anyway, I have one. Can you talk about a little bit the relationships, I mean similarities and differences between your approach and coevolution? Is it coevolution or it is something different?
No, I think it's a very simple, maybe the simplest way of implementing coevolution in somehow, but it's really we didn't want to find something to do something new. Something like this has already been done several times, but it was more or less a demonstration that we could perform quite extensive computations and tackle this problem in quite, let's say, from several points of view, with some reliable, for example, results, reference results, just mainly because of the lot of work we have done with the implementation of the algorithm by GPU. So actually, we are probably considering this more a demo problem than an actual field in which we wanted to do something new in the short term. But as we saw, there are quite a lot of open issues that could be worth exploring, and we actually got some, I think, some interesting responses.
Let's see if there is any question in the meantime. No, so let me ask another. You could find, in some senses, the ground truth with that method of exhaustive search of the parameters, because the parameters were 5. Yeah. And I am already impressed because it's already a very, very big job, I think, to find it. But if the parameters are more, you cannot do it. Yeah. So you are basically assuming something like, if the approach works for 5 parameters, it will work also for more. Yeah, so that's more or less the reasoning, right?
It was mainly to have some reliable, for this problem, some reliable ground truth. Of course, you cannot have it for much more complicated problems. But
okay, if there is no other question, we thank Stefano again. And we can switch to the last speaker. Is the last speaker here? Christian? The speaker is not here, I think. So, structure-based constraints in genetic programming, Christian. I'm sorry, the font is small and I am almost blind. Vechuis or something like that. No, the speaker is not here. So we anticipate the coffee break, I think. Okay, and we close the session here. Yes, let me remind you again that there will be another session of Alea after the coffee break, and that the closing session is at 6 and not at 6:30. Okay, so in two different points of the program, we have 2 different informations. In one point is 6, in one point is 6:30. The right information is 6. The closing session will be at 6. Okay, thank you very much.